Hello students, today I am going to teach you adrenal gland hormones. So let's discuss first adrenal gland in short. The adrenal glands are paired organs lying near the superior pole of the kidneys. It is also called suprarenal glands. The right gland is shaped like a pyramid, whereas the left gland is semilunar in shape and larger of the two. Together they weigh about 8 grams, but their weight and size vary with age and physiological conditions of the individual. Each gland has two concentric regions, a yellowish adrenal cortex and a reddish brown central adrenal medulla. The adrenal cortex has three concentric zones in which the cords of the epithelial steroid producing cells are arranged somewhat differently and which synthesize different classes of steroid hormones. So the first one is the zona glomerulosa. Immediately inside the capsule and comprises about 15% of the cortex. The steroids produced by this are called mineralocorticoids because they affect uptake of sodium and potassium and water by cells of renal tubules. The principal product is aldosterone, the major regulator of salt balance. Next zone is the middle zone, that is zona fasciculata. It occupies 65 to 80% of the cortex and consists of long cords of large polyhedral cells. These cells secrete glucocorticoids, especially cortisol, which affect carbohydrate metabolism by stimulating gluconeogenesis in many cells and glycogen synthesis in liver. The innermost zone, that is zona reticularis, comprises about 10% of the cortex and consists of smaller cells. Cells of zona reticularis also produce cortisol, but it primarily secretes the weak androgens including dehydroepiandrosterone, DHEA, that is converted to testosterone in both men and women. The innermost portion is the adrenal medulla, which is composed of large pale staining polyhedral cells arranged in cords or clumps and supported by a reticular fiber network. Medullary parenchymal cells, also known as chromaffin cells, arise from neural crest cells. Unlike cells of adrenal cortex, chromaffin cells contain many electron-dense granules. about 150 to 350 nanometer in diameter, for storage and secretion of catecholamines, that is either epinephrine or norepinephrine. The adrenal cortex and medulla can be considered two different organs with distinct embryonic origins, functions, and morphologic characteristics that became united during embryonic development. The cortex arises from mesoderm and the medulla from the neural crest. So now come to the receptors of glucocorticoids and mineralocorticoids. Glucocorticoid and mineralocorticoid receptors both are members of thyroid or steroid hormone receptor superfamily of transcription factors comprising of a C-terminal ligand binding domain, a central DNA binding domain interacting with specific DNA sequences on target genes, and an N-terminal hypervariable region. Glucocorticoid receptor reside primarily in the cytoplasm, whereas thyroid hormone receptors are located in the nucleus. So after ligand binding, the cytoplasmically localized glucocorticoid receptors translocate to the nucleus. So glucocorticoid receptor can also act at the membrane or in the cytoplasm to activate or repress signal transduction pathways, providing a mechanism for crosstalks between membrane and nuclear receptors. So most nuclear receptors bind to DNA as dimers. Consequently, each monomer recognizes an individual DNA motif referred to as a half site. The steroid receptors, including glucocorticoid, bind to DNA as homodimer, consistent with this two-fold symmetry, and their DNA recognition half sites are palindromic. So now coming to mechanism of action of glucocorticoids. The binding of glucocorticoid to the glucocorticoid receptor in the cytosol results in activation of the steroid receptor complex through a process which involves the dissociation of the heat shock proteins, that is HSP90 and HSP70. Following translocation to the nucleus, gene transcription is stimulated or repressed. Following binding of dimerized glucocorticoid receptor ligand complexes to a specific DNA sequence in the promoter region of target genes. So this is the basic mechanism of action of glucocorticoid receptor. So this glucocorticoid receptor element, that is GRE, is invariably a palindromic sequence which binds with high affinity to two loops of DNA within the DNA binding domain of the glucocorticoid receptor, and that is zinc finger motifs. This stabilizes the RNA polymerase II complex following, facilitating gene transcription. The interaction between glucocorticoid receptor and the two particular transcription factors are important in mediating the anti-inflammatory effects of glucocorticoids, and these two transcription factors are C-FOS and C-Jun, and they are subunits of activator protein 1. And activator protein 1 is a pro-inflammatory transcription factor induced by a series of cytokines. The glucocorticoid receptor ligand complex can bind to C-Jun and prevent interaction with the activator protein 1 site, thereby mediating the transrepressive effect of glucocorticoids. So similarly, functional antagonism exists between the glucocorticoid receptor and the nuclear factor kappa beta. So nuclear factor kappa beta is a ubiquitously expressed transcription factor that activates a series of genes involved in lymphocytes development, inflammatory response, host defense, and apoptosis. Apoptosis is nothing but programmed cell death. So glucocorticoids mediate their anti-inflammatory effect through several mechanisms. I am quoting the few. The inhibitory protein 1K kappa beta, which binds and inactivates nuclear factor kappa beta, and thus anti-inflammatory effect is induced. Second is the glucocorticoid receptor cortisol complex is able to bind nuclear factor kappa beta and thus prevent initiation of an anti-inflammatory response. Third one is both glucocorticoid receptor and nuclear factor kappa beta compete for the limited availability of co-activators, which include cyclic AMP response element binding protein, that is CREB, and steroid receptor coactivator 1. So this is the mechanism of action of glucocorticoids. Now what are the effects of glucocorticoids? So first, on general metabolism. Glucocorticoids increase blood glucose concentrations. So this is the basic effect of glucocorticoids on metabolism. In the liver, cortisol stimulates glycogen deposition by increasing glycogen synthase and inhibiting glycogen phosphorylase. But hepatic glucose output also increases through the activation of key enzymes involved in gluconeogenesis, principally glucose 6-phosphatase and phosphoenol pyruvate carboxykinase. In peripheral tissues, muscles or fat, cortisol inhibits glucose uptake and utilization. So this results to increase in blood glucose concentration. In adipose tissue, lipolysis is activated, resulting in the release of free fatty acids into the circulation. An increase in total cholesterol and triglyceride is observed, but HDL level falls. It causes insulin resistance and an increase in blood glucose concentrations at the expense of protein and lipid catabolism. On the other hand, glucocorticoids stimulate adipocyte differentiation, promoting adipogenesis through the transcriptional activation of key differentiation genes including lipoprotein lipase, glycerol 3-phosphate dehydrogenase, and leptin. So thus glucocorticoid promotes visceral obesity, and this may be due to increased expression of both the glucocorticoid receptor and 11-beta hydroxysteroid dehydrogenase 1 in the omentum compared with subcutaneous adipose tissues. In a skin and connective tissue, glucocorticoids inhibit epidermal cell division and DNA synthesis and reduce collagen synthesis and production. In muscle, glucocorticoids cause atrophy because protein synthesis is reduced. Glucocorticoids inhibit osteoblast function, which is thought to account for the osteopenia and osteoporosis that characterizes glucocorticoid excess. Glucocorticoids also induce negative calcium balance by inhibiting intestinal calcium absorption and increasing renal calcium excretion. So this also promotes osteoporosis. Glucocorticoids suppress immunologic response. In the peripheral blood, glucocorticoids reduce lymphocyte counts acutely. T lymphocyte count is more affected than B lymphocytes. By redistributing lymphocytes from intravascular compartment to spleen, lymph nodes, and bone marrow. Conversely, neutrophil counts increase following glucocorticoid administration. Eosinophil count rapidly fall. It inhibits monocyte differentiation into macrophages and macrophage phagocytosis and cytotoxic activity. Glucocorticoids reduce the local inflammatory response by preventing the action of histamine and plasminogen activators. Prostaglandin synthesis is also impaired through the induction of lipocortins, which inhibit phospholipase A2 activity. In the eye, glucocorticoids act to raise intraocular pressure through an increased aqueous humor production and deposition of matrix within the trabecular meshwork, which inhibits aqueous drainage. Long-term but not acute administration of glucocorticoids increases the risk of developing peptic ulcer disease. So this is all about glucocorticoid. In next class we will discuss mineralocorticoids and androgens. Thank you.